And the good news is that none of the patients has presented any arrhythmia during follow-up. Now, I did the first surgery now more than 4 years ago, and the last patient I operated on was 2 years ago. So we start to have a reasonable follow-up now. The second endpoint in terms of safety was tumor, reason why these patients had sequential imaging studies before and after surgery. And again, so far so good. We haven't found any new abnormality in any of these patients. A third endpoint in terms of safety was alloimmunization. We were not so much concerned about that because, as you can see, these progenitors are weakly immunogenic. They don't express class 1 and very little and no class 2. And actually, when we did mixed lymphocyte reactions in each of the patients, as you can see, the responses were very weak compared with what you get in the control when you expose allogeneic peripheral blood mononuclear cells to other PBMC. You see the responses of the patients which were very weak. And consistent with this observation, only one patient developed donor-specific antibodies at a very low titer and without any clinical consequences. And this was obtained at the cost of a 1-month immunosuppressive treatment with intentionally low doses of immunosuppressants. Now, ejection fraction increased over time, which might simply reflect the benefit of the concomitant revascularization. More interesting maybe is the fact that there was a trend for an improved systolic thickening of the areas where I put the patch, but I think we should be extremely cautious about that. At most, it might be an encouraging hint, but the study was not powered or designed to assess efficacy. So I think we should stick to, so far, to feasibility and safety. And I think this is the best summary of the trial, which was written by Dr. Miller in the editorial published at the time we published our own paper. Just an encouraging hint, at least supporting the safety of the procedure once the population has been adequately purified. So now, what are the perspectives? Well, I think there are two main perspectives. One is to follow up in this direction, that is to move on with transplantation of cardiomyocytes derived from pluripotent stem cells. And actually, there are a couple of trials now on the preparation around the world, both in North America and in Europe. All these studies so far share in common to target the transplantation of more mature cardiomyocytes than those that we have been using. And you know, there is still this discussion whether we should transplant early progenitors or more advanced cardiomyocytes. Actually, I don't have the answer. For different reasons, we picked early progenitors, but those who want to transplant more advanced cardiomyocytes might be equally right. Each of these protocols has its specificities. In some cases, cells will be injected, and in other cases, as we did, the cells will be combined with a tissue engineering construct. Now, I think this is a very attractive concept, conceptually, to remuscularize the heart with new cardiomyocytes. As a clinician, I'd like just to highlight a couple of challenges which I think should not be underestimated. The first, obviously, is a large-scale production, given the number of cardiomyocytes which have been lost and which are supposed to be replaced. But this is doable. With pluripotent stem cells, I don't think it's a major challenge. We had a bank of 800 million pluripotent stem cells, so this is something doable. I think the second challenge is more tricky, because we know that most of these cells tend to die pretty early after the procedure, and this is just one example among others showing the magnitude of cell attrition over a short period of time. So for those who want to generate a new myocardium, which will stay there forever, or at least for a long period of time, it's a real challenge to keep these cells alive. And so if we look at the protocols which are in preparation, and once again, I'm not talking about basic science or preclinical work with genetic engineering or things like that. I'm talking about the clinically oriented protocols. There are different approaches. For example, in Seattle, Murray's group is promoting a pro-survival cocktail based on cell conditioning. Others prefer to use scaffold, as we did, either the engineering heart muscle presented by Heschenhagen yesterday, or the cell sheet of the Duplanis group of Sawa with the iPS-derived cardiomyocytes. So this is another option, and I think it makes sense really to combine cells with some form of scaffolding. Now, maybe in the future, we'll have HLA haplotype lines, and I think we're going to hear about that in a few minutes, but we're not yet there. Insofar, for keeping the cells alive, we still have to rely on immunosuppression, conventional immunosuppression, and this is a challenge because you have to define the appropriate drug, the appropriate dose, and because we're dealing with patients who may not be so tolerant to a lifelong immunosuppression. They may have hypertension, renal failure because of their underlying heart disease, and so it is a problem. And I draw your attention to the fact that if you look at this macular degeneration trial in which ES-derived retinal progenitor cells have been used successfully to some extent, the only complications that have been reported so far are those associated with immunosuppression. And finally, there is this issue of arrhythmia, the prevention of which requires a couple of prerequisites, but this has been alluded to elegantly yesterday by Dr. Laflamme in particular. So I just, again, would like to draw your attention to the fact that you can achieve nice engraftment, as you can see with these extensive areas of green cells. And this is associated with an improvement in function, but in this particular study at the cost of an increase in early arrhythmias. That might not be a clinically relevant concern, but we cannot ignore that. And one issue here is if we look at the data with either the patches or the injections, it's the same. Either injections in the myocardium or epicardial patches, you always have this very thin layer of fibrosis insulating the graft from the host, which might make coupling more difficult than we think in the real world. Interestingly, in this particular study, there was a positive outcome in terms of function, but the same benefit regardless of whether the cells were alive or not. In other words, when they irradiated the cells were no longer alive, but you get a similar benefit, which tends to support the paracrine mechanism and takes us to the second perspective, which would be to use the pluripotent stem cells no longer for transplanting them directly, but rather for using their secreted products. Why that? Well, first of all, because there is compelling evidence now that most of the benefits of pluripotent stem cells or cells at large can be duplicated by administration of their secretome. And this has been shown in the heart by our group and others, but also in the lung, in the kidney, and in the brain. So there is fairly compelling evidence for this equivalence. Now, that said, to be efficacious, the secretome must come from mother cells, which should address two major characteristics. First, they need to be early differentiated because they have a higher secretory profile. And second, there is evidence that there should be a match, the phenotypic match. In other words, if you take the secretome of cardiac-committed cell, it works better than if you take the secretome of fibroblast or non-cardiac cells. And in this setting, pluripotent stem cells are very attractive because they tick the two boxes. You can drive them to make them cardiac-committed cells, and you can freeze the process at the stage you're interested in. That is, you can use progenitor cells. And we've shown that in a recent study where we derive extracellular vesicles from cardiovascular progenitors from iPS, used a mouse model of a chronic heart failure, and found, and this was a randomized blinded study, that actually the EV, the secretome, was associated with the best outcomes. Now, one step further, we try to figure out the mechanism by which function had been improved, use this fate mapping study. Again, this model of chronic heart failure in mice, but we failed to show, we showed an improvement in function and a reduction in infarct size, so that was great, but we failed to show the generation of new cardiomyocytes using these fate mapping studies. Conversely, we found evidence for a reduced fibrosis, increased angiogenesis, and in the subsequent set of experimental studies, interestingly found a decrease in inflammation, manifest as both a reduction in the infiltration of pro-inflammatory cells in the myocardium, as well as a decrease in the circulating levels of pro-inflammatory cytokines in the blood of these animals. So this definitely has some advantages for a given level of equivalence, and I just would like to highlight one of these advantages, which is the lack of immunogenicity. You can see here the profile, the immunological profile of the cardiovascular progenitor EV. You see that definitely they don't express class 1 or class 2, and actually we found that they failed to trigger any proliferation of T lymphocytes. They don't stimulate NK degranulation, and overall they seem to be immunologically neutral. Now, before closing, I'd like to briefly address the three major translational challenges, because once again, this was supposed to be clinically oriented. Three major challenges. The first is obviously the purification of the medium, and there are different techniques which are available. Probably in a clinical perspective, tangential flow filtration is the best suited for clinical applications, but the issue here is not so much to pick a technique. The real question is to what extent should we purify? And there is increasing evidence that we may not have to be too selective in the fraction that we want to retain, and maybe the whole secretome is what is necessary for getting a maximum benefit. This has been shown in this study where you can appreciate, once again, the equivalence between cells and the exofraction. But interestingly, the best recoveries are seen when you take the whole conditioned medium. Now, the second is the characterization. And as you can see, there are hundreds, thousands of components on board, so that I'm not sure that we should try to deconstruct the cargo, because probably all of these components, to what extent, need to be there. And this is a trend now not to be too selective. One additional advantage of taking the whole cargo is that the EVs have the unique advantage of allowing to shuttle the package into the cells without being degraded. Whereas if you take individual components, they may not get such an easy access to the cells. And finally, delivery. And again, from a clinical perspective, there are two options. Number one, the patient has to undergo heart surgery, which is good for me. And in this case, probably it's good to functionalize a biomaterial with the secretome to allow slow release and controlled delivery. This has been done elegantly in the study by combining the secretome with a gel. And again, you will see that you find this equivalence between endothelial progenitor cells and their EVs, but interestingly, you also see that you get a much better recovery when the EV are in a gel compared with what happened when there is just a snapshot injection in PBS. So this combination is probably promising. Now, the other possibility is that the patient does not require heart surgery, and in this case, I think it's worth exploring the intravenous route, which has been shown effective in this particular model of doxorubicin cardiomyopathy, as you can see here, with this improvement in fraction, ejection fraction, a reduction in fibrosis. Now, the mechanism by which the IV approach is effective is still not very clear, but if we assume that inflammation is a major component of advanced cardiomyopathies, it may make sense that an IV approach may efficiently tackle this inflammatory component, and that might account why we already have a couple of clinical studies reporting positive results with IV administration of cells. So to conclude, I think that the pluripotent stem cells, either embryonic or iPS, really offer unique possibilities, not only because of scalability, but because of this ability to really target the optimal stage of differentiation we're interested in. You can like them or not like them, but I think we should stop saying, well, they give tumors, because so far the safety record has been pretty good across a wide range of organs, the spinal cord, the heart, the eye, and now we have diabetes, and now we have Parkinson's, so we start to accumulate reassuring safety data, and possibly both safety and efficacy could be optimally combined if we consider these pluripotent stem cells not necessarily as the agents to be transplanted, but as ex vivo producers of a secretome supposed to recapitulate their protective effects. I'd like to acknowledge the contribution of all my clinical colleagues, as well, obviously, as those who really did the work in the lab. Thank you.
Thank you, Philippe. Jay?
Philippe, that's great. We have you as one pioneer in the field. It's the first cell therapy in human. Now it's about 20 years ago. So maybe I missed your early data for the pluripotent stem cell derived cardioprogenitor cell. So this is not the pluripotent stem cell derived myocyte, it's the progenitor cell. The concept of CPC can be vague, and we just need the one cell skip, pluripotent cell skip. Then we can cause the problem of the tumor genesis. So because the concept of the CPC is not clear, if all over the world, China, Korea, everybody does CPC transplantation, if the cell characteristic is not clear, then one cancer, one tumor genesis can kill the whole field. So I would like to hear from you to caution the people that it's not my side. My side, we know. The CPC, for example, maybe the pluripotent gene, OCT4, KRAS, they all disappeared during differentiation. These standards need to clearly convey to the community before the in-human project going on.
Well, I fully agree with you, and that's why maybe I've been too fast, but that's why I have said that there was safety provided the population was appropriately purified. That said, we have spent a lot of time for this purification process because eventually you can have some contamination of your progenitors by cells that have not responded to the cardioinstructive cues. We found an average of 97% of cells which were really defined as cardiovascular progenitors on the basis of the conventional markers. And obviously, our regulatory agency was only interested in the remaining 3%. And so we had to do the transcriptomics of these 3%. It turned out that they did not longer express pluripotency markers as well. They express a mix of genes. And so when we injected these negative population, SSA1 negative population in immunodeficient mice, none of them developed any tumor. But I agree that purification is obviously an important step. That said, I'd like to remind also that it's important to look at markers like OCT4 and NANOG and so on. But these markers are also expressed on mesenchymal stem cells, which are widely used clinically. They are expressed on umbilical cord-derived MSCs without apparently any major adverse event. So I think the expression of the marker by itself does not necessarily mean that the line will be tumorigenic. But again, and to answer your question, I agree, purification is a key step if you're not using terminally differentiated cardiomyocytes.
Any more questions? Yeah.
I'm not in the field, so some naive questions. The human trial is extremely important, but for the tumorigenicity, it's still the major concern for the embryonic-derived stem cell. From your protocol, you listed 4 days of induction using different growth factor, right? Maybe, yeah. So I don't know the details, but still, even you use some kind of a screen, the field, people will still have worries. 4 days, is it too short? Do we have better, we don't need that big population. Do we have a better control to minimize the danger of the tumorigenesis? So because embryonic stem cells has been in the field in a very active research for basic science and applications for so many years. So can you, give us more perspective of your prediction, your hope for how much we can count on using embryonic stem cell as a source of a cell therapy? Thank you very much.
No, I will certainly not make any prediction because I don't know. So I don't make any prediction. I think that pluripotent stem cells, at least those, well, first of all, the 4-day protocol was just designed to generate the population of progenitors we're interested in. I don't believe that if you extend the exposure to the cues, you may reduce tumorigenicity. I don't think so. I think it's completely separate. That said, I think we should be very open-minded because honestly, we don't know. So pluripotent stem cell or their differentiated derivatives are among the players. You also have mesenchymal stem cells, and we have a couple of ongoing trials, in particular the DREAM heart failure trial with 600 patients. We should get the results next year, and maybe that will be an important step for knowing in which direction we should go. So I think that so far there has been no evidence, no conclusive evidence that a cell was better than the other one, except that probably there is a trend for using cardiac-committed cells. With the caveat that, again, mesenchymal stem cells seem to hold a promise. So I would not make any prediction. I think we just have to wait for the outcomes of the ongoing trials and see what happens regarding the outcome of the patients. But definitely, I still believe that these pluripotent stem cells will have a place, whatever it is, but they will have a place in the armamentarium of cell therapies that we may use in patients. And again, regarding tumors, I remind you that we now have a spinal cord injury trial with more than 4 years of follow-up. We have a macular degeneration trial. We have our own heart trial. And you have also trials now starting in diabetes and Parkinson. And so far, there has been no red flag.
Last question.
But even cardiovascular progenitors, they represent a very heterogeneous population of different cardiomyocyte subtypes. They would represent atrial, ventricular, nodal cells. And perhaps one of the explanations for the arrhythmogenicity of the transplanted cells could be the presence of nodal cells that could act as a focus for arrhythmia. What are your thoughts on that?
I think it's quite possible, but this is also one of the reasons why we skipped the use of intramyocardial injections and use a patch-based delivery. Obviously, this is only possible when you are a heart surgeon and have control over the heart. But I think that regardless of the phenotype of the cells that you are injecting, the fact that you puncture the myocardium in multiple sites with a syringe, needle, or a catheter is by itself something that can cause arrhythmia, regardless of the phenotype of the cells. This has been shown with different cell types. That said, it's clear that the population may harbor cells with a potential for automaticity. But again, I think the patch-based delivery is a convenient way of addressing this issue. And again, all our patients had a defibrillator. They had sequential, countless numbers of EKGs, and we never saw any arrhythmia. And this has been the same experience with Sawa in Japan with shell sheets of myoblast. When they injected myoblast, they got arrhythmia. When they used myoblast as a cell sheet, as a patch, there was no arrhythmia in their clinical trial.
Thank you, Philippe. Might move on to the next speaker now, who's Keiichi